Assalamu alaikum guys and welcome to my YouTube channel Biology Articles and MCQs. Guys, our today's topic of discussion is a life cycle of HIV. But first of all, we will briefly discuss about the HIV. That HIV is the human immunodeficiency virus that is the main cause of AIDS. So we will discuss in this video that how it will enter in the body of the human being and how it will cause the AIDS. But first of all, we will learn about its step or stages. That a different step or stages of the life cycle of HIV are as follows. The first is attachment, the second is fusion, third reverse transcription, the fourth will be integration, fifth will be transcription, and then assembly, and then last will be budding. So let's discuss these all as 7 steps or stages in detail. Here you can see the life cycle of the HIV, human immunodeficiency virus. First of all, let us discuss that what are these things. First of all, this is the cell, and this can be human cell, and this will be specifically the white blood cell of or leukocytes, because the HIV will attack the specifically white blood cells in the human being. And first of all, this is the HIV virus, and this is the CD4 receptor. Guys, there are specific places on a specific cell that will enter the virus to it. So, the specific place that is present on the white blood cells of the human beings that will help in the attachment of the HIV is the receptor that is called as CD4 receptor. So let's suppose that this is the HIV virus, and this virus is now first of all will attach to the specific sites that are CD4 receptors on the white blood cells. So this is the first step of the attachment of the attachment of HIV virus on the CD4 receptors of human leukocytes. Guys, you know that this is a simple structure, and here is the envelope of the HIV virus, and inside the envelope there is a protein coat which is also called protein capsid. And in addition to this, the HIV also contain the heredity material in the form of RNA, and the RNA will be in the form of two identical strands. So these black ones are the RNA, and these are the two identical strands of RNA. And these circular portions are the specific enzyme that is called reverse transcriptase enzyme. So this is the brief structure of the HIV, that it consists of an envelope and a protein capsid, and the heredity material is in the form of RNA. Two identical strands will be present, and the one enzyme is present that is also called reverse transcriptase. So the first in the first step, the HIV will get attached to the CD4 receptor, and after the attachment, it will fuse with the cell membrane. After fusion, the protein capsid and the hereditary material plus reverse transcriptase enzyme will enter into the cell, and the envelope of the HIV will not enter into the cell. So here you can see that these are the virus RNA that has entered into the cell. After entering the virus RNA into the cell, it has to convert into the DNA. So, in order to convert the virus RNA into DNA, one enzyme is required, that is the reverse transcriptase, and this reverse transcriptase enzyme is present inside the HIV virus. So, it will convert the RNA into DNA. This reverse transcriptase enzyme will help in the conversion of RNA into DNA. Yes, first of all, the RNA is converted into one strand of the RNA is converted into one strand of DNA, and you know that the DNA has ability of replication, so it will replicate, and this strand will act as a template, and after replication it will form a double stranded DNA molecule. So now that RNA is converted into the DNA molecule, and because this process is the reverse of transcription, so this process is also called reverse transcription, and the enzyme required here is reverse transcriptase for the conversion of RNA into the DNA. So this is the second step of this is the third step of the reverse transcription. Here reverse transcription, the third step is takes place. Now this double-stranded RNA will enter into the nucleus of the cell. First of all, you have to remember that this process of reverse transcription takes place in the cytoplasm of white blood cell. But after converting into the DNA, it will enter into the nucleus of the white blood cell. And here it is entered into the nucleus. You can see that this is the main DNA molecule that has entered. And after entering the viral DNA inside the nucleus, the viral DNA will integrate or mix up with the DNA molecule of the white blood cell. So here this red part is shown that this is the viral DNA, and this remaining portion is the DNA of the white blood cell. So this is the fourth step, that is integration. Fourth step of integration means the viral DNA is mixed up with the DNA of the specific white blood cell. So now guys, this is because this is the life cycle of HIV, and we need many copies of HIV from here. And so the HIV has the main hereditary material is in the form of RNA. So this DNA should be converted into the RNA first, so so that it will become the part of the next the HIV, newly formed HIV. So here will be the transcription will goes on, and you know that the process of transcription converts the DNA into the RNA molecules. So this DNA, that is hybrid DNA, because it contains some copy of HIV and some copy of the host cell. So it is hybrid DNA. Hybrid DNA is transcribed into the RNA molecule. So this is the fifth step of transcription. After converting into the RNA molecule, after undergoing transcription, it will come out of the nucleus again into the cytoplasm. And in the cytoplasm, the RNA will undergo translation, and you know that in the translation process, there is a formation of proteins by the help of RNA. So the RNA is now converted into the different proteins. After converting into different proteins, now the main requirement of the hereditary material, that was RNA for the HIV, and the proteins are now synthesized. So these protein and RNA will assemble together, and after assembling, it will assemble in the form of main HIV. So this process is the sixth process, or the assembly, or you can also call it as reconstruction. And after assembling of the total HIV virus, it will again go into the infuse with the cell membrane of the host cell, and after fusing into the host cell, the fusion will be in the form of budding, and this bud is formed here. And this is the seventh step of the process of budding. After budding, there is the formation, or there is the release of the newly formed HIV virus. So this is all the process that is about the life cycle of the HIV virus. I hope you understand all about this cycle, and don't forget to share this video with your friends. Thank you so much.